All right, so this video is on male reproductive tract anatomy and endocrine control. And I'll apologize ahead of time for the cursor. There's a lag between me trying to move it and it actually moving. So try to keep that in function, working. And so what I want to do is start out by sort of going over essentially the plumbing of the male reproductive tract, how things are connected one to another, and then we'll get to the point where we talk about production of spermatozoa, the male gametes, and how that process is under endocrine control, as well as the development of secondary sex characteristics also under endocrine control. So we have in this image a sagittal section of the male pelvis. We can see the reproductive structures. And if we talk about the production of sperm and then collectively the production of semen, what sperm are suspended within, we have to look at the testis down here. Right, so we're going to show you in a subsequent slide sort of the structures within it. It has a tubule system where the sperm are developing, and actually it also has cells that are producing testosterone and estrogen. Its tubular system is connected to this system of tubules known as the epididymis. This is a highly coiled, it's about 20 feet long, highly coiled tube that leads into the vas deferens, this tube right here. The vas deferens is going to come up bilaterally, right, one from each testis, enter the abdominal cavity, and through the inguinal canal. And again, you'll see that better in another image. And then enter the urogenital tract at what's called the ejaculatory duct. And so the vas deferens seen here, and then we'll come up sort of behind this, deeper in this image to the side of the bladder, and then enter here, which is this tubule is called the ejaculatory duct, entering the prostate gland, one of the accessory glands in males. And the ejaculatory duct connects with the urethra, right? So here's the bladder up here, the urinary bladder, and here's the prostate gland. Here's the ejaculatory duct meeting the urethra. So that's where the urogenital tract and the reproductive tracts sort of combine or come together in the male. If we follow the pathway here that semen containing sperm would contain, it's going to pass through the urethra. Actually, there's segments of the urethra. There's the prostatic urethra. And then through this wall of muscle, it's called the urogenital diaphragm. There's the membranous urethra right here, this little short segment. And then we're in the penile urethra, sometimes called the spongy urethra because it passes through the corpus spongiosum of the penis and then exits here, right, at the external urethral orifice. Along the way, we have not just the prostate as an accessory gland, but we have the seminal vesicles up here. Those are paired. Prostate is a single. There's only one of those. The seminal vesicles are paired. There's one on each side. And then here's the bulbourethral gland, sometimes referred to as the Cowper's gland. And those three glands add secretions during a process called emission, which forms semen. And those secretions contain a variety of substances, lubricating substances for the urethra of the male as well as the female reproductive tract, buffers to buffer urinary acids, antibiotics, prostaglandins that are contractile to both the male and the female reproductive tract, a clotting factor essentially that will cause the semen to sort of coagulate in the female reproductive tract. And so a whole variety of essential, another one, fructose, right, provides nutritional support for the the sperm cells, which really don't store any nutrition at all, and so require require nutritional support from their environment. And so those sperm cells are added to the semen that's being produced by these three separate groups of accessory glands. And the secretion of those three accessory gland structures, sorry to keep moving around with the cursor, but it keeps it from freezing up. But the secretions of those three accessory glands is supported by testosterone, and even probably more significantly by dihydrotestosterone. It's called DHT, right? Abbreviated DHT. And we talked about that before, right? That's the peripheral processed version of testosterone, at least at some locations, where testosterone is converted into DHT, and the DHT then has the primary effectiveness at those target tissues. And the accessory glands are one of those tissues where DHT is a more prominent activator than testosterone. Okay. So if we blow up the testis and the epididymis, as well as the vas deferens up here, we start to see this sort of tubular nature of the male reproductive tract, right? So the testis itself contains these little tiny tubules in here called seminiferous tubules. There are about a half a mile worth of seminiferous tubules, all along which the spermatogonia, the stem cells that will produce sperm, are dividing and developing, right? And so at any given, there's a cycle of production, and at any given, in any given seminiferous tubule, there are multiple sites where sperm cells are maturing at a given instant. So while it is cyclic overall, in other words, the production of sperm at a location is cyclic, the fact that there are probably hundreds, if not thousands, of locations where that cycle is taking place means that sperm are produced essentially continuously throughout the reproductive lifespan of a male. And that is quite significant. We're talking about 25,000 sperm per minute. And that contrasts sharply with females who produce one viable egg, or ovulate, I should say, one viable egg or oocyte a month, right? So 25,000 per minute versus one per month. There's quite a dramatic difference in sort of the reproductive strategies of gamete production in males versus females. So anyway, these developing sperm move out of the seminiferous tubules into this structure called the rete testis, and then finally into these ducts called efferent ductules. And what those are connecting to at several locations is this long convoluted tubule called the epididymis. It doesn't look like a single tube in this image, but it is. It's just highly coiled up. And the sperm are still maturing while in this epididymal tube where they spend about a week. By the way, in the seminiferous tubules, it takes about 74 days to go from stem cell to the sperm that leaves the seminiferous tubules. And then about another week in the epididymal tube, which is about 20 feet long, highly coiled. And then finally into the vas deferens. And the vas deferens can actually hold sperm for quite a while, upwards of a month, and they remain viable. They're pretty much fully mature and motile by the time they leave the epididymis, sort of down here at the tail. They leave the epididymis to enter the vas deferens. The vas deferens is sort of like a holding space, if you will. Its wall is very muscular and is involved in the process of ejaculation, or the expulsion of the sperm from this tubule into the ejaculatory duct that we saw in the last picture, and I think we see in the next picture as well. Okay, so here's my cartoon version of some of the same things. Right, here's our highly coiled seminiferous tubules. Remember, about a half a mile of these. We'll blow those up in a minute and show you the tubule and where the cells are found in a second. They're going to drain their sort of partially developed spermatocytes into this rete testis, which then goes to the efferent ducts and then into the epididymis, right? Again, for further maturation for about a week, and then into the vas deferens, right? Where they're going to hang out for as long as a month and still remain viable. Remember, the vas deferens will pass through the abdominal wall, right, at the inguinal canal, right? So down here we are in the testis, right? The epididymis is in the testis. The vas deferens goes through what's called the spermatic cord and passes through the abdominal wall. And remember, here we come into the prostate at the ejaculatory duct, right? One from each side, obviously. And then we're here in the urethra, and we can sort of see the prostatic ducts sort of emptying their contents. So the prostate is where the seminal vesicles, shown up here, and the prostate would add their secretions to the sperm coming from the epididymis through the vas deferens, right? And then, of course, the Cowper's glands add their secretions last, buffers and lubricants to form the sort of composite secretion known as semen. Okay, if we blow up the wall of a seminiferous tubule, we can see that it has this sort of structure where the germ cells are on the outside, right? And then there's this layer of these darker cells called Sertoli cells. In general, they're called nurse cells. Whoops. Sorry. In general, they're called nurse cells because they're helping with the maturation of the developing spermatocytes. So just the shorthand version here is that the spermatogonia, the stem cell, will divide. One of its daughters will remain as the stem cell. The other will sort of pass into the wall of these Sertoli cells. They actually, the Sertoli cells have cell-cell connections that sort of zip and unzip, if you will, as the cells are passing between them. Shown, this is my cursor wakes up. Shown here. So here are the cells sort of passing between the Sertoli cells, and they're connected more than I'm showing them here. And then the not quite mature spermatocytes, spermatogonium, are released here into the lumen of this seminiferous tubule. And again, those will be carried off out of the testis into the epididymis. Now, I think I hinted there a second ago, the next slide just shows a histological view of this, and then we'll do a blow up of these Sertoli cells and what they're doing. Okay? Okay. So here we have that blow up. So here's the outer wall of a single seminiferous tubule. Remember, the spermatogonia, the stem cells are on the outside layer. And then it's hard to pick them out, but the Sertoli cells are sort of these big blob-like cells just inside the stem cells. And then the developing spermatocytes are sort of within that Sertoli cell wall. And the Sertoli cells form what's called the blood-testis barrier. And what that means is that they're maintaining an environment inside the seminiferous tubule that's dramatically different than the environment outside. And that's essential to spermatogenesis, the production of these spermatids and then eventually sperm, right? The viable gametes. Now, it's important to point out, because it's going to be really important in terms of the endocrinology, the Leydig cells are the secretory cells that respond to LH stimulation that are found outside the seminiferous tubules, right? So this is actually where our testosterone secretion is going to come from, and we'll show you a slide of that coming up. Okay. So here we have, again, going back to sort of the cartoon version of the same thing, right? The outer wall, the seminiferous tubule. Here's our stem cells, the spermatogonium. This big guy with all the arms sort of sticking out to the sides is that Sertoli, quote-unquote, nurse cell that's sort of zipping and unzipping these connections. If you can see them here and then over here, right? So the developing spermatocytes are sort of passing through this blood-testis barrier in a controlled fashion. And one of the things that's happening along the way is dramatic mitosis. And so this original division, right? Remember, 25,000 per second. Yes, it's happening at a lot of locations, but at each location, we need to be making a lot of spermatozoa. And the only way to do that is through lots and lots of mitosis before we get to the meiosis, right? Going from diploid to haploid. Anyway, so here we are again at the inside of that tubule at the lumen where the loose spermatozoa are going to be released. And again, they're not fully developed yet. They're sort of anatomically developed, but they're not fully functional yet, as again, that occurs in the epididymis. And then again, once again, here's our Leydig cells on the outside of that tubule. Okay, so here's our Leydig cell. Let's focus on the Leydig cell for just a minute, and then we'll combine how that Leydig cell is interacting with the Sertoli cells to sort of promote spermatogenesis. So our Leydig cell is responsive to LH, luteinizing hormone, again, named for its action in females. But it is the hormone that stimulates Leydig cells through cyclic AMP, right? So a G protein coupled receptor there. And it's going to stimulate the metabolism basically tied to this cholesterol metabolizing enzyme that's going to lead to a whole cascade of steroid like structural transitions from pregnenolone to progesterone to testosterone. And then actually to a degree through this enzyme called aromatase from testosterone to estrogen. So this aromatase enzyme is really important in females because their primary reproductive hormone is estrogen, although you could throw in progesterone as well. Whereas in males, right, at least in terms of circulating levels, it's testosterone. However, it's important to point this out, right? Males are sterile that lack significant aromatase function. In other words, estrogen is absolutely required in males for, down here at the bottom, spermatogenesis. Okay, now just to back up a hair, right, and we'll see this in the next slide, LH and FSH, the other, its sort of counterpart hormone, come from the anterior pituitary, right, and they are stimulated by GnRH, which came from the hypothalamus. And so it is the GnRH pulses. Remember, that's one of those hormones that has a very pulsatile release pattern. And the rate of those pulses determines this sort of degree of LH and FSH secretion, and actually the balance between LH and FSH, which one is secreted more or less than the other. If we follow our testosterone being produced by the Leydig cell, it's going to, let's follow maybe the more essential function first, is it's going to be taken up by seminiferous tubules. Remember, this is a lipophilic hormone, actually, and the estrogen. They're both lipophilic hormones, will be taken up by the seminiferous tubules to promote spermatogenesis, one of the key elements essential to spermatogenesis. Without testosterone, the spermatogenesis stalls in meiosis 1, and you essentially produce no sperm. So that would be characterized as paracrine because we're diffusing right from outside the seminiferous tubule to inside the seminiferous tubule. They're right adjacent to each other, and so we don't need to be carried in the bloodstream. However, testosterone and to a lesser degree the estrogen produced also end up in the bloodstream, where we have many distant target tissues. I just mentioned previously the reproductive gland tissues, right? The accessory gland tissues, the prostate, the seminal vesicles, the bulbourethral glands. But we could also talk about, skeletal muscle being a target. We could talk about bone. Remember, we talked about bone mass and reproductive steroids before. Hair, body hair, distribution and stimulus of that. Sebaceous gland activity. There's a whole variety of target tissues. And again, often acting through conversion to the dihydrotestosterone. At least it's some of those target tissues. Okay, here's where we're going to combine the actions of LH and FSH and the Leydig cell over here with the Sertoli cell of the seminiferous tubule over here. And so we just saw a moment ago here's our GnRH pulse coming from the hypothalamus, stimulating the anterior pituitary to release LH and FSH. The LH acting on the Leydig cell. Don't forget, we said this before when we were talking about pituitary early in the semester. Prolactin was an essential hormone for maintaining expression of the LH receptors, right? And so that tied prolactin into male reproductive function. Our testosterone and estrogen produced, again, they can go off into the bloodstream, but here we have them being taken up. They're lipophilic, so they pass right into the Sertoli cells. Now they could diffuse right back out, right? But we want the concentration both inside this cell and in the lumen of the seminiferous tubule to remain very high. To do that, we need to trap it once it's diffused. Both of these, either one of them have diffused into this space, into this cell or this space. And so we need, especially, and again, this is especially true for testosterone, we need an androgen binding protein. And so that is actually stimulated by FSH. So here we are following the other half of the pituitary hormones. The FSH target is the Sertoli cells. Again, cyclic AMP is our messenger here. Stimulating transcription translation to increase the production of androgen binding protein. Well, once our testosterone binds to it, it can no longer diffuse back out and into the bloodstream, right? And so what that does is it increases the testosterone concentration. Notice this androgen binding protein is also being secreted, but it's going to increase the concentration of testosterone in this space by about 100-fold higher than it is in the blood. And so again, yes, this is a small space, but the only reason we can keep such high concentration here is by trapping it. And the only way we can trap something that's lipophilic is to stick it to something that's not lipophilic, in this case, the androgen binding protein. And then, of course, our androgen binding protein testosterone complex is what's going to promote spermatogenesis, along with that little bit of estrogen that's being produced through conversion from testosterone using this aromatase enzyme. Okay, and here we have sort of a histological slide of the exact same thing, right? Here's our wall, our developing sperm occurring here, right? In this region, if you can follow my cursor, right, we would have the Leydig cells, right, sitting out here in the sort of the interstitial space, so outside of the seminiferous tubules. And over here on the right, we can see those developing spermatocytes passing through this pink Sertoli cell, and then finally being released into the lumen, which would be up here at the top. All right, I think that'll do it for males.